Today I am going to discuss about sterilization. Hello, I am Pavankumar, B.Pharm second year, Maharajah College of Pharmacy. What do you mean by sterilization? It means the killing of microbes. Why we have to kill the microbes? Because to prevent the contamination on sterile products, to prevent the decomposition and spoilage of food and food products, to prevent the contamination of unwanted microbes in pure culture and other microbiological experiments performed for research studies, to prevent the unwanted microbial contamination in antibiotics, enzymes, vitamin fermentation and other industrial processes, and to prevent the contamination in aseptic areas which are used for the preparation of the sterile dosage form. How can we what are the methods for the sterilization? There are two methods, physical method and chemical method. In physical method, the sterilization can be done by using some equipments like hot air oven, autoclave and radiation, and the chemical method is by gaseous sterilization and by using disinfectants. We are going to discuss about the physical methods of sterilization. In that, dry heat sterilization, sunlight and drying and flaming are included. In dry heat sterilization, most reliable and rapid method of sterilization. The killing of dry heat is due to the protein denaturation, in which the microbes are killed by the denaturation of protein in them, and the microorganisms are more resistant to dry heat as compared to the moist heat, and therefore this process requires higher temperature and longer exposure times. As we are familiar with the sunlight and drying, as sunlight possesses ultraviolet rays which along with the heat rays are responsible for the appreciable germicidal activity. This is the natural method for sterilization of water in tanks, rivers and lakes. And flaming, the article is passed over the flame without allowing to become red hot. Example for that is the mouth of culture tube, glass slides, needles and cover slips, etc. And moist heat sterilization. The sterilization by moist heat means the killing of microorganisms with hot air or steam. The lethal effect of moist heat is due to the denaturation and the coagulation of proteins. Moist heat sterilization is divided into three forms: the temperature below 100 degrees, temperature at 100 degrees and temperature above 100 degrees. The temperature below 100 degrees, in which the heat liable fluids may be disinfected by heating at below temperature 100 degrees, and a temperature at 100 degrees is that the boiling at 100 degrees for 10 to 30 minutes kills all vegetative and bacteria and some bacterial spores, and temperature above 100 degrees, as which the saturated steam are more efficient and sterilizing agent, that is, for example, autoclave. And the filtration or the mechanical method and the radiation or cold sterilization are also included in the physical method. As in the filtration or mechanical method, this is a non-thermal method of sterilization used widely in pharmaceutical industries, where the heat liable solutions are to be sterilized. The following types of filters have been used for sterilization: asbestosis filter, also known as SIDS filter, sintered glass filter, filter candles. These are normally used for the water filtration purpose as filter candles, and membrane filter. These are also known as millipore or ultra filter, and the radiation or cold sterilization. The use of ultraviolet rays, ionizing radiation, X-rays, gamma rays and the beta rays. That's all it for today's topic, and thank you.